Dear students, dear audience, ladies and gentlemen, good morning. First, it's my pleasure to be here. I was having another program for this Sunday, but I think I have made the right choice. I appreciate being here. In my presentation, I'd like to talk about some aspects of the immunology which go beyond the traditional interpretation of immunity having something to do with the protection of the body. So I'd like to convey the message that we consider as an immune system has many other far-reaching possibilities and potentials. Perhaps it's going to change the landscape, how we are looking at the immune system. Over the past couple of years, the immunology has undergone some major changes in terms of the spectrum, how it tackles with the maintenance of the human and the human tissue integrity and body integrity. I'd like to talk about the certain aspects, how those tissues that are particularly responsible for the defense, these are referred to as peripheral lymphoid tissues or secondary lymphoid tissues, are organized, and what is the implication, how they are organized. Also, what we consider as a spectrum of lymphoid tissues seems to be expanding. And I'd like to point some territories which have escaped attention and probably have a major impact in maintenance of body integrity. As any other complex system, if we can refer to something of a pointer that has stopped functioning, but anyway, I'm trying my best. So it's a Murphy's law that anything that can go wrong will go wrong. So we have different outcomes. We hope for the best, that the immune system will operate in a normal functional physiological way, but in many cases it won't. And the last one is some kind of a summary and giving some hopes that perhaps the immune system can be tackled in a more efficient way than it can be today, but there are also some hazards. So what I'd like to point out at the beginning is that there is an English word called prolepsis, meaning a word which is presuming or expecting something to happen in anticipating it and tackling it. Okay, so preemptive defense, so to speak. Next. So I switched from red to green. Fine, that looks better. Thank you. So the essential part is that our immune system is born with us. It is not induced by any kind of exposure. What does that mean? It means that even in our mom's womb, our system prepares for the postnatal period. Why would the system do so? None of the immune organs are essential tissues. So why are we born with lymph nodes, spleen, and the other things? What predicts and what predisposes us to have such organs? So if you just go to the literature and look at the verbalism, the words, or the descriptions that were there in 1980, if you just type you just go to the Medline and type in the word immunology or immunity, you get something over 20,000 hits. And if you add immunology and tissue and development or regeneration, the orange and the gray letters are just minuscule compared to the total hits. Now, if we skip 40 years and we look into the same search words, we find a substantial increase, like fourfold increase, or maybe even almost fivefold increase, but more than tenfold increase when it comes to immunology and tissue and development, and also a substantial increase when it comes to regeneration, which is another form of tissue development. So it reminds me to this statue that you happen to see in Dublin in the National Archaeology Museum, which is a very interesting statue. It's like 2,000 years old. And you would think that it is two faces. No, it's three. The last one is poking to behind. So to see the whole thing, you need to walk around and appreciate the complexity. So what I like to talk about how the immune system builds up, with particular emphasis on the secondary lymphoid tissues, that we need new tools, new possibilities, new avenues to tackle these. We just heard one possibility before with the artificial intelligence. But when it comes to wet lab, then we certainly need new antibodies and reagents, specific new reagents, new instruments and procedures, very complex, a complete science unto its own. Absolutely important is the capacity to perform multi-parametric analysis, by very advanced bioinformatics. And when it comes to human health, obviously we have to prove that our hypothesis works, so we have to use mouse or any other animal experimentations. In our work, we've been involved mostly in the first part to produce, and in the last part, to produce new specific monoclonal antibodies with which we can tackle the complexity of the immune system, particularly of the cells and the tissues. And here, what you can see here are just a few slides from a mouse spleen. You can appreciate the T cells are in turquoise and the B cells are in magenta. They seem to be nice two complementary parts of the white part of the spleen. Now, if we throw in one more monoclonal antibody, which stains a particular subset within the follicles, this is the B cell zone-rich area, you can see that these fibroblastic-looking cells are here, but they are not in the T cell zone. So in addition to having the leukocytes segregated to separate parts, the underlying tissue also segregates accordingly. So one type of fibroblasts are living along with the B cells, another set lives along with the T cells. And if we throw in two more monoclonal antibodies, again, two macrophage subsets, then we can see that this edge region around the white path is also very complex. We have two layers of macrophage subsets. So this is really necessary, this kind of even higher resolution, to dissect the complexity of the lymphoid tissues. Now, when it comes to doing research, we have to have some rational models. Over 90% of the animal kingdom are actually non-vertebrates. So when it comes to immunological research, the typical approach is to use mouse or rat or some other models, but they are representing less than 10% of the total. And even within this less than 10%, the overwhelming majority is built around mouse models. So there's this huge bias in what kind of models, in vivo models, we are using. And this is pretty much restricting element. So, but we make some kind of prediction that they are probably representing the same. Why? Because over the past 60 million years or so, we've been coming along with the mouse. Now, but as this is very much restrictive, and actually it still allows us to make outside this box fundamental observations and discoveries, is reflected in this list. I appreciate for being so small, but the list is so long. Okay? These are Nobel Prize winners, including our guest of honor, for discovering the GFPUs, and also Gilles Hoffman, who discovered in flies the impact of what we call the pattern recognition receptors, which are now a complete new and very important direction even in human immunology. And while we are in Szeged, my old friend István Andó has made great discoveries and seminal observations in Drosophila immunity. So here locally, you guys can be proud of that person who has made seminal discoveries in immunology. So what are the common themes in the lymphoid tissue development? I hope you can appreciate, without knowing what these antigens are, is that in the lymphoid tissues, there is an overwhelming majority of lymphocytes that are visible, okay, and other forms of leukocytes. They have some kind of tendency to accumulate selectively, but within the lymphoid tissues, they seem to occupy different territories. So T cells, now described as this kind of silverish, grayish cells, are separate from the B cells, and the B cells are gathered around this specific stromal subset, so non-lymphoid subset. Now, irrespectively whether this is a spleen or Peyer's patches or lymph nodes, they have the same principle. Now, as to the development, however, we have different developmental principles operating behind the formation of these tissues. The spleen is a single organ which has a very peculiar switch of functions. While we are in our mother's womb, it functions as a hemopoietic tissue. It's like a bone marrow, okay, but in your belly. But it's not bone marrow. The other form or system that evolves gradually are the gut-associated lymphoid tissues, with particular emphasis on the Peyer's patches. Now, they have a kind of segmental orientation along the length of the intestinal tract. And the last bit, which is also a multi-organ-like distribution, are the lymph nodes. But lymph nodes do not evolve randomly. They always have the same location. So that means that they are developmentally programmed, okay? Now, they have a very interesting sort of order of appearance. They start at head place, and they are sort of progressing towards the tail. Now, if you look at the spleen first, it starts, lo and behold, as a part of the stomach. this is a 12.5 days old mouse embryo. This is the stomach cross-section. You can see the lumen, the cavity, and this protrusion is the spleen. Gradually, it evolves, and in a matter of 2-3 days or so, there's a vast expansion and increased vasculature. So it's not just leukocytes that are gathering there, but it is a vascular supply that also has to expand. Now, this is a critical period because if we look at the spleen, what you can see here, various abbreviations, these are transcription factors, so DNA binding elements that have to have a certain hierarchy. That's two sets, two parallels. You can see here the upper and the top mechanism, and there are upstream elements and downstream elements, and any of those, if are abrogated, then they will block this spleen development. On the other hand, there'll be nothing wrong with the lymph node or Peyer's patches. So they will serve something similar, their structure will be something similar, but the developmental path is entirely different. Now, if you look at the lymph nodes, then I know it looks a somewhat haunting image because it's a bit too overcrowded, maybe too complex, but just pay attention to the center here, where we have some pre-adipocyte cells in a close association with some neuronal cells. These neuronal interactions are needed to prime some fibroblastic elements to interact with some leukocyte subsets. And with the contribution of the lymphatic endothelium, they start to become specific cells that only at the lymphoid tissues will prevail, okay? So it's not the lymphocytes themselves that will make up the lymphoid tissues, but at least 3 other cell types together. So it's a kind of convergence of different cell lineages. And the last element is that when there is a sufficient amount of T and B cells, so mature lymphocytes, they will transform the local vasculature to make high endothelial venules, so such venules that will have a high capacity to recruit lymphocytes. Okay, so it's a positive forward loop to amplify those cells that will support the existence of lymphoid tissues. The mucosal lymphoid tissues, like the Peyer's patches, have somewhat a similar principle in appearance, a kind of in between the two, but they are more likely similar to the lymph nodes. Now, when it comes to the gut, however, the other aspect that my title refers to is the time. By the time of the birth, we are okay with the spleen and the lymph nodes. But importantly, the gut-associated lymphoid tissues need to mature afterwards. This is a secondary wave, if you like, of differentiation, and that refers to those sites that are there in an immature form, but they need some kind of microbial contamination, so to speak, to evolve. So in other words, we have a certain set of lymphoid tissues we are born with, but when it comes to antigenic exposure, they can expand. Now, I don't know how you guys are university students, but trust me, being a university student is full of frustration and tensions. So sometimes students just wish if their head and brain would grow. And when it comes to lymphoid tissues, they do grow, okay? Huge. And then, but what happens afterwards, we don't know. Certainly, they have a capacity to multiply several-fold in size, and then to regress. And one of these transformations is seen here, where we have a small gathering of lymphocytes, lymphoid cells with certain marker expressions. B cells are missing. But when it comes to some kind of contamination, they evolve into these singular organisms or organs called isolated lymphoid follicles. So this is the pre-exam period brain, and this is how I wish it to be. Okay, when it comes to passing anatomy, physiology, and biochemistry, preferably the same day. So our research was very much motivated by the appearance of the spleen and the development of a spleen for several reasons. In the late 1990s, that was an absolutely shocking discovery that one single gene called lymphotoxin alpha, if deleted, was sufficient to block the formation of the lymph node and the Peyer's patches. This is dramatic because in immunology there is some degree of redundancy. So if you lack one cytokine, then maybe 2 or 3 other can compensate for it, but not with this guy. So lymphotoxin alpha deficiency on its own is sufficient to block the vast majority of not just looking good, but simply being there, okay? Now, in contrast to that, there was a parallel observation indicating the importance of this transcription factor family. This is a normal spleen. You can see the difference between the T cells and the B cells. Now, if we have a lymphotoxin beta receptor mutant mouse, the spleen is there, but you see some kind of a chaotic distribution between the green and the red cells. And pretty much a completely different scenario occurs if we have a deleted gene for this particular transcription factor, and you can see the macroscopic differences. There's a normal spleen and two mutant ones. They look absolutely strikingly different. So we're interested in what's the difference, what makes the difference. And to our greatest surprise, what we found is that if you look at a normal spleen, and then you have the T cells and B cells and some specific stromal elements, but you don't see this particular type of vessel, which is very characteristic for the lymph node. Now, in the absence of this transcription factor, the spleen will start looking like this one, so it's basically full of ectopic lymph node-specific vessels. So I hope you understand. It's like switching. I know maybe there is some mostly neuro guys here in this audience, but it's like making your brain look like spinal column. It's a vast difference. And in addition to that, the spleen never has any kind of lymphatic vessels. You may remember that the lymph node development does depend on the lymphatic vasculature, but not the spleen. So when it comes to using the GFP, again, courtesy of our this year's guest of honor, this is a lymphatic endothelium-specific driven expression of GFP in the spleen. You never get to see that in a normal spleen. So it seems like a complete reversal of vasculature, making it look like a lymph node. Okay, so it looks as though vascular patterning is a critical parameter for defining immune organs. Now, if I had asked you before this lecture, just list a few cell types that would contribute to the immune system, I pretty much doubt that you would have mentioned endothelial cells or blood vessel cells. But they are inherent part of the story, and it looks as they are critical part of the story. Now, what else do we know that we don't know yet? Okay, that lymph nodes, Peyer's patches, spleen can filter and search for antigens. But what is interesting is that you have other anatomies, like body cavities, abdominal cavity, pleural cavity, pericardial cavity, that seem to be unprotected, which is just foolish. Those tissues need to be protected, and with completely different research, we ended up with finding these organs in the mouse belly, adipose-associated tissues, which seem to be collecting both normal lymphocytes, and what is the bad news, they can also collect malignant lymphocytes. So we haven't had the chance to look at it in humans, but we seem to have some adipose-associated tissues that are preferential target size for malignancy. Not to become cancers, but to accommodate cancer cells, okay? Now, we have named these particular organs as foliate lymphoid aggregates because of the flag-like appearance, and they seem to show some kind of a similar T-cell, B-cell segregation. As you can see here, the red guys here are the T cells, and the green guys are here the B cells. Not as complete as the lymph nodes or the spleen, but somewhere halfway. And the last bit I want to talk about is not the birth, but the decline and associated pathologies. I just want to summarize real briefly that there seems to be a parallel. If you compare this side, then it indicates that the majority of the parameters or features are associated with decline of the immunological potency, okay? Elderly people cannot be fully vaccinated, or if you're having difficulties with full vaccination, and there are many underlying reasons why this is the case. But at the same time, elderly individuals have the capacity to have an augmented capacity to evolve, to initiate autoimmune diseases, okay? And this is actually logically contradict what I've just said, that with elderly, we have a declining immune system. Now, we have preserved capacity to establish extrafollicular or extra lymphoid tissue neonate or lymphoid neogenesis which can be very much associated with certain autoinflammatory and autoimmune diseases. So just a brief summary of my presentation is that perhaps we can consider the immune system is more just providing immunity. It has a fundamental role in tissue building, and how the tissue is preserved very much reflects on the immunity. So the two things are very much correlated. Also, if it comes to appreciating the whole immune system, we have very severe limitations. Why? If any of you guys have had to go for medical checkup for some kind of indication of immunological abnormality, what was the sample? I guess it was your blood. Certainly, you didn't get your lymph nodes, tonsils, Peyer's patches, everything biopsied for considering some kind of an indication of abnormality. So we have a very severe limitation of what is accessible. So why do we do that? Why do we do research on immune organ development, lymphoid tissue development? Because it's beautiful to start with. It's a fantastic imagery that you can make, and then appreciating how a very complex system where half is stable, sessile, it won't ever move, and the 90% will always move, they complement each other. And in that hope, we hope that we can find alternatives for going into the tissues. We can find alternative diagnostic methods and tools, and perhaps that can be translated into research, and hope we will be able to do that before ChatGPT or artificial intelligence would kill the scientific literature. So thank you for your attention.
Thank you very much for this very interesting but a little complex lecture. It went very well. And now I am wondering if there are questions from the audience for the presenter. There have been lots of issues addressed very nicely. Okay.
So I have two questions actually, and they are rather about the autoimmune diseases. Firstly, I would like to ask if there are any proofs that the immunological memory plays a role in the progress of them. If there's any information about that.
I congratulate you for your bravery to ask the first question, and I apologize for me being deaf. Could you please? I got something about the immunological therapy with immunological memory, but I cannot make out the connection.
Okay, so my question was that if there are any information about that, if immunological memory plays a role in the progress of autoimmune diseases.
Oh, a very good question. Immunological memory is typically a product of a very complex immune reaction, where the highest survivor, the most efficient cells have to mutate, particularly the B cells, that have to have the highest affinity immunoglobulin production, which is a completely random process. You can consider it as a Russian roulette. And in some cases, this kind of mutation goes awry in a way that instead of being specified to the eliciting antigen, it switches to something reactivity against some self-component. So yes, immunological memory has a basic role in establishing highly aggressive and very, in many cases, untreatable autoimmune diseases. How do we know this? Because typically the high affinity autoantibodies are IgG type immunoglobulin, and this is a characteristic of memory type immune responses. So yes, the two things are correlated.
Thank you so much, and I have another question, that how do autoimmune diseases affect the homeostasis of the immune system?
I would say because one is what we see, it's an observational thing, that what we see in the elderly is that the original very broad spectrum of different antigen specificity is very much narrowed. And instead of having, I don't know, just number, 10 to the 12 or so different clones, we'll have 10 to the power of 8, something. So a substantial reduction and a compensatory expansion of the residual space. The lymphoid tissues are defined in a physiological space. And the lymphocytes, particularly the T cells, can sense the void. With other words, if there is no other lymphocyte, then they can expand, okay, irrespectively of the antigen being present. So we think that the composition and the clone homeostasis very much influence what the immune system is capable of. So with the memory type immune responses, with any kind of antigen stimulation that involves a defined space, I need to point out that the immune responses do not appear in the blood. They need tissue, which is a defined and sort of limited anatomical territory. So there's a competition. The lymphocyte world is extremely crude, no mercy, okay? If there is no step-by-step acquisition of stimulatory survival signals, then the cells will be deleted. So the fittest, the best will survive. And in many cases, it can be at the expense of the physiological normal cells. And the last bit that just occurred to me, during the aging, it's not just the antigen specificity that changes. There are new types of cells appearing. One relatively recent observation is that there is what's called aging-associated B cell, ABC, okay? Like ABC killers in the Quran. So the aging-associated B cells, on one hand, seem to be exhausted B cells, do not want to do too many things, but the only thing, or many things that they can do is produce inflammatory cytokines, and in many cases, autoantibodies. Okay?
Okay, thank you so much.
Any other question from the audience? If not, then we have to hand over the certificate of appreciation, and we will move forward with the program thereafter.